In this short series of videos, we're going to be looking at how to measure how quickly or how slowly evolution occurs within a population by looking at how the genetics of that population change from generation to generation. Now, this seems like a fairly complicated concept, but actually when applied to looking at a specific phenotype, it's actually relatively simple to understand. So let's get the terminology out of the way first. Now, the first concept that we need to look at is the concept of what a gene pool is. Now, a gene pool is simply a look at all of the different possible alleles, that being different forms of the same gene, that result in different observable phenotypes within an organism's population. So in the case of this frog population here, the three different phenotypes that we can observe are green, red, and purple when it comes to the skin color of these frogs. Now, once we've identified each of the different alleles, each of the different forms of the same gene, and if we calculate what the total population is, we can calculate what the allele frequency of each different phenotype is going to be, which is simply the percentage of which a specific allele occurs within the total population of the organism being studied. For example, if we use skin color of this frog population here, the first thing that we need to do is identify what the total population is. So 2 plus 3 is 5, plus 2 is 7, plus 3 is 10, plus 2 is 12, plus 3 is 15. So if we were to calculate the allele frequencies of each of the different phenotypes that we see that red is the least frequent allele, which occurs with a 1 in 15% chance. We can see that purple is the second least frequently occurring, that being in only 2 out of 15 members of the population, and green is easily the dominant allele, which occurs in the majority, 13 out of 15, and that's simply what allele frequencies are. Now, if we were to track these allele frequencies, that being what percentage of our frog population is green, red, or purple in 1 generation from now, or in 2, or again, theoretically 10 generations down the road, we can see whether any evolutionary change is occurring, because we can see whether one of the three phenotypes is being favored by a specific selection pressure. Now, in order to understand this concept, we need to talk about the idea of genetic equilibrium, which is exactly the opposite of the situation that I described. Now, whenever we see the word equilibrium in any type of science, this refers to a state of constancy, and meaning that genetic equilibrium is simply when all of the allele frequencies being studied within a given population remain constant or relatively constant. And we can take a look at a beetle population here. So let's say that we are looking at the phenotype of exoskeleton color here, and this time we've simplified. There are only 2 different alleles, green and yellow. Now, in the ancestral population, or the first generation, if you will, we can see that 75% of all DNA codes for a green exoskeleton, while the other 25% codes for yellow. If we look at the observable phenotypes here, we can see that 50% of our population is homozygous for green, and the other 50% of our population is heterozygous for both of the exoskeleton colors. And then if we look at the next generation down the line, we can see that even though we don't have 6 beetles anymore, we actually have doubled our population for a total of 12, but we can see of those 12 beetles, we can see that 6 out of 12 is homozygous for green, meaning that this allele frequency remains basically identical. The only thing that is different is that we now have a homozygous yellow beetle here, which throws off the 50% split from being exact, but the allele frequency itself at the genetic level remains almost identical between the offspring generation and the parent generation. Now, if this is true, if the allele frequencies, the frequency of the population that has a green exoskeleton and a yellow exoskeleton remains identical from generation to generation, that means that overall our population is not undergoing any specific evolutionary change because neither phenotype is favored over the other. And this is what we call, in population genetics, this is what we call the concept of a genetic steady state, which is basically a synonym for genetic equilibrium, which simply states that the allele frequencies, the frequencies of which each phenotype occurs within the population, remains constant, or at least very close to it, unless a specific selection pressure is applied within the ecosystem in which this population lives. And once again, several examples of selection pressure might be increased competition for a specific resource. For example, a food shortage or a water shortage would increase the competition between members of the same population. An increase in the number of predators would be another example of a selection pressure, because only the organisms with the phenotype more suitable towards escaping or evading predators would survive. Another example would be the presence of a disease that favors one phenotype over the other. This would be another possible selection pressure here, and you get the idea. Now, in the event that a selection pressure occurs, this would disrupt, theoretically, the frequency by which a specific phenotype occurs within the population. But we're actually going to deal with this second. Now, if the selection pressures within a specific ecosystem remain constant, meaning that they do not affect or do not prefer one phenotype over another and remain basically constant within the population's ecosystem, the resulting changes that occur to the allele frequency occur much slowly and much more gradually compared to if a selection pressure were strong. And for the longest time, this was believed to be the most common way that evolutionary change occurred, which is what we call the gradualism model of change, in that if the selection pressures that are applied to this given population remain relatively low and relatively constant, this is not going to affect the allele frequencies in any specific way. The problem is, if we consider what selection pressures are, do selection pressures always occur predictably and always occur at common rates? Well, if you think about this for more than 5 seconds, the answer, of course, is no. For example, are these beetles always going to have an abundant food supply? That would obviously be no. Is the predator population going to remain completely constant over the entire course of these beetles' lives? Again, that answer is no, and this unpredictability becomes even more obvious when we factor in quote-unquote natural disasters like disease, drought, or fire, or other environmental selection pressures such as that. Now, in the event that a very strong selection pressure occurs, we get another pattern of evolution, which is what we call punctuated equilibrium. And this diagram that we have here is actually a very good summary of how punctuated equilibrium occurs. So if we go to our common ancestor population here, we have a relatively constant phenotype variety or relatively constant allele frequency that occurs at this specific time. But then at this divergence here, we get the strong application of some sort of selection pressure that favors some sort of evolutionary change or some sort of evolutionary divergence, that being one or more specific phenotypes that are favored or that give the population an increased level of fitness compared to others, and therefore we have a relatively quick, by comparison, evolutionary change. And then if we apply a different selection pressure at this divergence here, we get again a relatively quick, at least in an evolutionary scale, divergence of a population into what are now two different species here. And again, what this selection pressure is simply depends on the environmental conditions that exist. These selection pressures could be an outbreak of a specific disease that favors one population over another as far as survival goes. It could be migration of a new species of predator in that is more easily able to hunt down and kill members with a specific phenotype over another. We don't know exactly what this is. It depends on the population. But the general trend in punctuated equilibrium is when genetic equilibrium is in existence, when we have these steady state conditions that exist, the rate of evolutionary change is very, very slow. In fact, there is almost no evolutionary change that occurs, because if there are no selection pressures, that implies that the population is well adapted to being able to find resources and reproduce without any particular trouble. But when a strong selection pressure is introduced to that population, it forces evolutionary change to occur relatively quickly and over a relatively short period of time, simply because that strong selection pressure is going to favor one particular subset of the population with a specific phenotype with a greater evolutionary fitness of being able to outcompete the other for resources, select appropriate reproductive partners, or simply survive whatever the natural catastrophe is going to be, resulting in much faster evolutionary change than perhaps occurred before. In the follow-up video to this video, we're going to look at the actual mathematical way that allele frequencies can be determined within a population, and how evolutionary biologists use allele frequencies in order to determine whether a population is in genetic equilibrium or not, and therefore predict whether a population is more likely to undergo gradualist evolutionary change or change according to punctuated equilibrium.